Profe.
Thank you very much. Yeah. Yeah. Okay. Yeah. I'll wait for Bill, who just went down the hall.
Okay.
I think you should all feel free to, I guess some of you, you'll start up here because you got computers, and then...
They're going to speak up. They'll speak up there.
Yeah.
I'll probably just speak here.
Speak from here.
Well, no, I don't
Yeah,
know.
up to you.
Yeah.
We had 75, 80 acceptances, so they'll drift in. Good morning. I'm Mike Van Dusen, Deputy Director of the Woodrow Wilson International Center for Scholars. Lee Hamilton, our President and Director, regrets he could not be here. He is at an all-day meeting of the President's Foreign Intelligence Advisory Board. It is my pleasure to welcome you to this important Directors Forum, the title, Stem Cell Controversy. In September, the Woodrow Wilson Center launched a new global health initiative. The initiative builds on the good work being done in many of our 21 programs at the Center to advance the dialogue on several key public health challenges of the day: the impact of health on development, the role of national and international institutions in global health policy, the spread of infectious diseases, and the emerging health technologies. Today, we are pleased to have with us three leaders in the field of public health who will consider a hugely promising and controversial emerging health technology, the use of stem cells. Dr. William Hurlbut is a physician and consulting professor in the program of human biology at Stanford University. He has done extensive work in biomedical ethics and was appointed to President Bush's Council on Bioethics in the year 2002. Dr. Michael West is President, Chairman, and Chief Scientific Officer of Advanced Cell Technology of Worcester, Massachusetts, which focuses on medical applications of cloning and human embryonic stem cell technologies. He is also Adjunct Professor of Bioengineering at the University of California at Berkeley. And Dr. Robin Cook, who will say a few more words about his fellow panelists, is a familiar and valued face here at the Wilson Center. He joined the Woodrow Wilson's Board of Trustees in 2005 and has provided essential guidance and direction to our efforts on public health. We're deeply appreciative of his leadership in putting together this panel discussion today. Dr. Cook is a physician and noted author. Indeed, his name is a familiar sight on the bestsellers list. His first novel, Coma, created an entire new genre of literature, the thriller based on medical issues. In books like Outbreak, Mutation, Terminal, and Toxin, he has thrilled and educated his wide readership, and several of his books have been made into movies and television productions. Dr. Cook's efforts have shone a light and advanced the dialogue on many pressing public health issues. Today, we are pleased that he has joined us to moderate this discussion on stem cell research. Please join me in welcoming the remarkable panelists. I turn the floor over to Robin Cook.
Thank you, Mike. I was particularly glad to hear him use the word literature in relation to my work. I don't get that up in Boston too often with the other institutions, including my own. I'm only going to say a few words because the two people that we have here are wonderful speakers. And if I thought about two people I'd like to get stuck in an airport with for a 12-hour flight delay, these are two individuals that would qualify for that. I thought I'd just say a couple of words from the sort of three hats that I'm wearing here. The first one that Mike alluded to of being involved here with this Woodrow Wilson Center, and that is how much I think that the Woodrow Wilson should move in the direction of technology and medical policy, which is what he was talking about. And I think this should be a good beginning. And I remember coming here in the fall and hearing a talk on the avian flu, which I actually wrote a book about 15 years ago. I'd like to also make a couple of comments about being a physician and why this issue is so important. One of the first things that you learn when you get to medical school, a big disappointment actually, is that you don't get a chance very often to cure anybody. I suppose that's a little bit like lawyers going to law school and realizing there's no such thing as real justice, or people going to the Kennedy School of Government and realizing that politics isn't quite what they thought before they went. That actually happened to me, by the way. But as a physician, you realize that most of the time all you're doing is putting your finger in the dike. And most of the illnesses and problems that you see with your patients are degenerative diseases. And I, for one, as a physician, suddenly realized that the potential of this stem cell is that there would be an opportunity to actually cure people. And this is tremendously exciting, I think. And the other points I wanted to make is from the point of view of being a novelist. It's going to sound a little like an infomercial, but I wrote a couple of books on the stem cell debate. One in particular that I mentioned just very briefly, it's called Seizure. And it's a book about the unfortunate politicization of this issue. And the message, of course, in that book is that I don't think politics is the right arena for deciding what to do in medicine. And I have a feeling, and in fact, at the back of that book, I talk about the fact that I think that the English have come up with a better solution to dealing with these problems, these emerging problems, because research in the reproductive arena is just picking up speed now. Instead of approaching it on an ad hoc basis, it would be better to have some sort of nonpartisan committee, which the English did form. But I think that I believe that at least one of the panelists here don't share my views that would work here in this country. But nonetheless, I thought it was something that would be worth trying. One of the reasons I wrote the book Seizure about this issue is because I was really amazed how few of my friends, who I consider very intelligent and informed people, really didn't even understand the issue. They understood the debate per se, but they didn't really understand the issue. In other words, that they thought that it's a little bit too complicated or something that they wouldn't be able to understand it, but that's not true. Biology has become very complicated, kind of interesting progression when you think about the 19th century being chemistry and the 20th century being physics, and now the 21st century seems to be biology, and biology is becoming extremely complicated. But some of the central points are really not that complicated. And one of the things that I thought that was very interesting, and again, we'll have a little disagreement here, is about the embryonic stem cell issue, is that I thought that the term itself was unfortunate in that embryonic stem cells are not taken from embryos. They're taken from a structure that has yet to become an embryo. When I studied embryology in college and also in medical school, it was very clear that embryos didn't start developing until around the 21st day. And prior to that, it was called something else. And in fact, most of the cells of that something else went on to develop ancillary structures that made it possible for the embryo to develop and then to succeed in becoming an individual. But even that is sort of interesting, that if embryonic stem cells don't come from embryos, it does sort of affect the argument in some people's minds. The other thing I realized a lot of people didn't realize is why was there such a huge hubbub about this? And they didn't realize that the real reason is that it brushes up against the most highly politicized issue in the United States, and that's the abortion issue, in which the people against abortion want any activated cell, that is an egg cell that's either been fertilized or activated by nuclear transfer or even more mysterious process of activating itself. they want that to be labeled as an individual so that it has full constitutional rights, et cetera, et cetera. That's the argument. And of course, that's the reason that this has become such a divisive issue. Because these little cells are being taken from something that could go on to develop into an embryo. Well, with that said, I'd like to introduce the first speaker, and this is Mike West. I met Mike West one time by just calling him up. This is when I was doing research for a book I was writing about this, and I just became... I was so interested, and just to listen what he was saying, what they were working on, and also something about his background, which is very surprising. I'll let him discuss that if he feels like it. But he is going to talk a little bit about where this research can go and its potential. Mike?
Thank you, Robin. It's a pleasure to be here and to talk to you today about the stem cell controversy. I'm here today. I'm going to try to discuss in as dispassionate as a basis as I can both the opportunities and the challenges of this emerging field we call regenerative medicine. I'll be showing you some slides that come from our biotech company. As Robin mentioned, I'm an officer in advanced cell technology, and as part of that, we're a public company. I'll be showing you certain forward-looking statements, and so take a safe harbor in those projections. So, embryonic stem cells. Let's start at the beginning. Let's talk about what they are and what all the hubbub's about. Why is the scientific and medical community so excited about these cells? Some people have drawn parallels between these cells and the biomedical revolution that occurred back in the late 70s, the recombinant DNA debate. And the excitement about these cells derived from several things. The most widely written about characteristic of these cells is the first bullet point I show you here. They're the first human cell ever cultured in a laboratory dish that can transform itself into every cell of the human body. So we call them a totipotential stem cell, unlike any cell that ever been cultured in a laboratory dish before. And as far as we know, different from any cell that resides in an adult human body. So there are no cells like this in us. They're an evanescent cell that exists for a few days, as Robin mentioned, before life begins to develop from this cluster of cells. They're a primordial, blank, unformed cell from which life can arise. And that unique property is what makes them, one of the reasons that makes them exciting. A second reason, which I'll try to discuss since we're having an informed intellectual effort here today in trying to inform everyone of the full potential and challenges associated with these cells. A second important characteristic, which is not often discussed, is these cells have a unique property, which I'll describe, which allows them to be genetically modified in any way. And so not only are they blank and able to make everything in the human body, that we can genetically engineer these cells in any way. We can take, now that we have the sequence of human DNA, we can take a T nucleotide that resides maybe in the gene that encodes hemoglobin in the blood cells and change it to a C, and then make anything in the human body, including potentially blood cells. That is a unique property of these cells as well. And of course, as Robin mentioned, the opportunities of using these cells for human disease is very promising. There are many challenges associated with these cells as well. There have been people who have pointed out not a single human being has ever been cured from a disease with these cells. And of course, the reason is they're brand new. It would have been impossible for anything to have been developed yet. But still, there are challenges before us. They make a very complex mixture of cells when they begin to differentiate. We have yet to learn many of the ways of inducing them to become many of the cells that we need clinically. And individuals like myself in biotech need to work out all of the quality control procedures and standard operating procedures, and we need to deal with an issue called histocompatibility. The cell lines that currently exist would be rejected probably by most patients because of foreign tissue rejection, and I'll touch on that in my little talk. And lastly, we need to identify practical applications of these cells. As Robin mentioned in describing what these cells come from these clusters of cells that are called pre-implantation embryos. Some people don't even call them embryos, as Robin mentioned. Others do. Whatever we call them, they're a cluster of cells surrounded by a halo of cells, again, as Robin mentioned, that will become placenta. And this cluster of cells inside has not yet begun to develop. They'll begin to develop about a week after this blastocyst would attach to a uterus and start a pregnancy. And the concept was, about 10 years ago now, that if we could obtain blastocysts from IVF clinics and isolate this inner cell mass and culture it in the laboratory dish and then allow these cells to replicate, we might be able to capture what is called a cell line, a stable growth of these cells that we could then turn into cells needed for the treatment of disease. On the right, what looks like maybe an island or something, that's about 1,000 human embryonic stem cells, one of the first lines that President Bush addressed us about back in 2001. This is one of the original now registered cell lines for NIH funding. Now, as I mentioned, these cells are exciting to cell biologists and medical researchers because they're a stem cell, but they're the mother of all stem cells. If we turn this diagram upside down, this might look like the branching of a tree. These cells are at the very trunk or the base of the tree itself of human cellular life and can branch out into inner ear hair cells for potentially for treatment of deafness, every cell in the human body. And this is nearly magical to a cell biologist. We're in the few short years that these cells have existed, we're already beginning to understand how to make many useful cell types and learning how unique these cells are. Here we see some pictures. On the right you can see neurons branching out, talking to one another. This is, again, unique property of embryonic stem cells. When they make neurons, they form synapses very easily, and they tend to want to form complex neuroassociations, which would potentially be useful for the treatment of course, diseases like spinal cord injury or degenerative diseases in the brain. Now, you remember I mentioned these cells have a unique property besides their ability to make every cell in the human body. And here is where potentially we see not just the ethical controversy associated with these cells, but another misunderstanding. These cells have a unique property that we in the scientific community call replicative immortality. These cells have the ability to replicate without limit, and we believe the reason for that is they're so primitive. They, in a sense, they don't think, of course, but if they could think, they may be thinking, look, I may become muscle, I may become bone, but you know what? I might become sperm or egg. And if I become sperm or egg, I need to then make another baby and another human, and then make more sperm and egg cells. And so I may be part of what's called the human germline, the reproductive lineage of cells that bridges the generations and has replicated since the dawn of life on Earth. A profound thought. And so we believe these cells are so primitive, they maintain that replicative immortality of the germline. It doesn't mean they can't be destroyed. It means that they can replicate without limit if properly fed and given nutrition. So here is a colony of these cells growing on mouse feeder cells, which of course have contaminated the cells, but nevertheless, on the diagram on the right, I'm showing you they have this property of immortality. Why is that important? It's important not because it implies that using these cells would make people immortal. That's not my point. My point is if we culture these cells in the laboratory dish in the undifferentiated state as these stem cells, they will replicate without limit. That then allows the confluence of molecular biology, the biology of DNA, with cell biology, and it allows us to potentially genetically engineer these cells without limit. Targeting genes, removing genes, changing, moving DNA around. And that confluence of these two currents of endeavor, molecular biology and cell biology, is a profound one, and it's one of the important reasons these cells are important. Now, as I said, this field is glowing with opportunity. It's also has a lot of challenges, and it's one of the reasons we've asked for, been proponents for federal funding through the National Institute of Health. An example of where additional work is needed is in the culture of these cells free of any known pathogens. The cells I showed you were growing in the presence of mouse cells. What in the past was a required condition for their growth. We and others have now published papers showing that it's possible to grow these cells. This cell line H9 is one of the registered cell lines. But on panel B, you can see these cells are now growing on these feeder cells that are from the ES cells themselves. And so if the cells themselves are clean and we can characterize them as being free of any virus or anything, then the feeders, of course, would be as well. Well, the problem is, of course, any research, federally funded research on these cells is currently impossible under the current guidelines because these cells were not derived prior to 2001. The additional research, of course, needs to be done on these cells in a broad array of areas, one of which is how to derive particular cell types that are needed medically, and is just an indication of sort of the power of these cells to differentiate. I'm showing you here that these cells can even differentiate into complex tissues, not just different cell types. In the second panel, A, you can see a cross-section through intestine that was formed from embryonic stem cells. And the red halo you see perhaps around the lumen of the intestine is smooth muscle that normally would contract intestine. That's just an example. In panel D, in the lower left, you see a hair follicle growing, having been made from embryonic stem cells, which many people would argue is a more significant medical application than any other. Now, so in a short discussion like this, it's hard to talk about all of the opportunities and challenges, but a significant one that abuts this issue of biomedical ethics and policy making is the desire of many in the research community to solve the problem of rejection. If we had learned to differentiate these cells into all the medically useful cell types that we needed and had funded that through the NIH, we would still have a problem with transplant rejection because the lines that exist today, if they were used to make, let's say, beta cells for diabetes, they would probably be rejected by most patients since the body rejects foreign cells and tissues often. And what has attracted the interest of the medical research community and worldwide, really, is to try to adapt the biology of nuclear transfer that was used in the cloning of animals to the cloning of stem cells, but not the cloning of people, an important distinction. And so if these little dots here represent cells, and the yellow cells represent the cells of the body, the somatic cells, and the blue cells, the germline, somatic cell cloning, instead of taking, I think I have an arrow here I can use, instead of taking reproductive cells to make more individuals, cloning is a technology which allows us to take a somatic cell, a cell from the body, and return it back into an egg cell. And as Robin mentioned, this transforms the DNA of a somatic cell in ways we do not completely understand back to the beginning of life, allowing us to make an embryo that could potentially generate a cloned animal. What occurred to us is, what if we use this technology in humans, but not to clone humans, to take a somatic cell back on a ride through the germline to reprogram it, but then make just a single lineage, potentially, from the human body, never a developing entire human being. And that's called therapeutic cloning. And I'm going to talk briefly about that. This is how it classically works. If we take an egg cell, here are the chromosomes glowing in blue. We remove them with a micropipette. This is a difficult procedure because, of course, this is surgery on a single cell. So it's not as easy as it looks. Here is a somatic cell floating in the same dish. This is a skin cell, an entire cell. And if we take it up in the same little manipulation pipette, and we can introduce it into the realm of the egg cell, we give it a little tap here to put the cells in physical contact, and then not to bring these cells alive, but to fuse the two cells, we give them an electric shock. And that fuses the two cells together to make one, and then the egg cell takes over and reprograms the somatic cell. Now, as we said, what we would then do is take the reprogrammed cells and generate embryonic stem cells and make then lineages of cells that might be needed by the patient. That would be called therapeutic cloning. If we transferred an embryo into a uterus to start a pregnancy and a developing human or animal, that would be reproductive cloning, and that's an important distinction, we think. The ethics debate, which we'll be discussing today, is, well, but is this itself a cloned human being? And this is, of course, the matter of debate. The excitement of the medical research community is that, of course, if you bring these technologies together, which is commonly called regenerative medicine, we now have potentially any kind of human cell genetically engineered in any way, but otherwise having a nuclear DNA genome identical to the patient, which you would predict help us solve this problem of transplant rejection. We have a fair amount of data. The critics of this technology often say, well, but show me the evidence that this will work. We have a fair amount of evidence now in animal models, both bovine and murine models, that this works. Here's, for instance, skin cells from a cow. Here's the clone blastocyst, here's the inner cell mass, and here's ES, embryonic stem cells. So we had turned, a skin cell into embryonic stem cells, and we've subsequently shown that we can make many different types of cells and transplant those cells. An interesting and provocative aspect of this, perhaps, is that as amazing as nuclear transfer sounds, and maybe regenerative medicine in general sounds to the ear, in my mind, it's even more wonderful than all I've already described. We now know that important causative factor in some degenerative diseases of aging is caused by what we call cellular aging, or the erosion of DNA at the ends of the chromosomes. They're called telomeres, these little lights on the ends of the chromosomes. So cells in our body shorten telomeres like a burning fuse. They age. And the remarkable thing is nuclear transfer in all of our animal research actually resets telomere length. So here is the lifespan of a cell. We took cells at the end of their lifespan. You can see the curve had flattened out here, and used these in nuclear transfer experiments and showed that we could reset the cell lifespan through nuclear transfer even beyond what they were before. What am I saying? What I'm saying is nuclear transfer, when we transfer a body cell back into an egg cell, not only does it reset the DNA to be an embryonic stem cell, take it back to the beginning of life developmentally, it resets this clock of cellular aging as well. So what we can make is cells with the full replicative lifespan that they had originally in animal models. This has never been demonstrated yet in human. Numerous animal species, though, this works, including the first clone mouse. This is Cumulina, the Teru Wakayama's first clone mouse. Then he cloned Cumulina. So these clones here are clones of the clone, and then he cloned the clones of the clones. And he showed in a paper that the clock of aging got repeatedly reset every time he performed cloning. As I said, in the few short years we've worked in the field of regenerative medicine, we actually do have, for the amount of time and the amount of capital that's been available for researchers in this field, I think a significant amount of data that support it. We've demonstrated that nuclear transfer-derived cells in animal models, again, were not rejected when we transplanted them. Here you can see engineered kidneys that we made in collaboration with Harvard Medical School, and here's urine in the sac. So this kidney was not rejected because it was cloned, and this was rejected because it was not. As Robin said, this area has been controversial, and so we and others have looked at other ways of solving the problem of transplant rejection, one of which is called parthenogenesis, the activation of an egg cell without ever seeing a sperm or without ever doing nuclear transfer. The only published results on this, these are actually human parthenotes. The only published data on this are the appearance of ES-like cells as primary cultures. There is not, as of today, there still is no published paper showing stable ES cell lines made by parthenogenesis. Now, another criticism is, no one's ever demonstrated any medically useful cell types yet from embryonic stem cells or nuclear transfer-derived embryonic stem cells. Nevertheless, I would argue we are fairly far along in the early days of this. These are pigmented cells we noticed early on in the dish. Scientists at our company, Irina Klemenskaya, noticed that these might be cells from the developing eye, the retinal pigment epithelium, that we believe may be causative in an important age-related degenerative disease called macular degeneration. So if we could make these very primitive cells, shown this patch of cells here, we could potentially transplant them into the eye to help prevent the complications of this disease. That blinds, it's the most common cause of blindness in the elderly. Well, these cells are relatively well characterized to this date, so we've shown that these cells gobble up little particles like the RPE cells should. They have certain markers here. You can say we're staining these cells for certain markers that are unique to retinal pigment epithelium. These are published papers. And the human ES-derived retinal pigment epithelium have more genes in common with the normal early fetal retinal pigment epithelium than any of the lines that we've studied that currently exist, including a well-known RPE cell line called ARPA19. And so in many ways, these human ES-cell-derived retinal pigment epithelial cells may have immense promise for diseases like macular degeneration. It's too early to tell for sure. These cells are currently being tested by us in animal models. There are other important applications which we and others are focusing on. There's a cell called the hemangioblast that can become blood and vascular endothelium and may have important applications in diseases like myocardial infarct. The largest cause of death in the United States, of course, is heart disease. Here we made cloned hemangioblasts that we labeled blue, and when we injected them on each side of a heart attack in a mouse model, we saw a band of regenerating heart muscle you can see here in this photograph in the damaged heart. We're also looking at genetically engineering such cloned and genetically engineered hemangioblasts to target the growth of tumors so that when a tumor recruits blood vessels to grow, they will have these cells that are sort of a Trojan horse that have entered the realm of the tumor to be induced by X-rays, perhaps a beam of X-rays to induce the local necrosis of the metastases. So there's a lot of interest worldwide. As part of this debate is the worldwide interest in the lack of maybe federal funding, which is now being taken up by states and other countries. The complicating this field, of course, is the debate about ethics, and Dr. Hurlbut will next, I think, address some of the ethical issues here. The debate is over the moral status of this entity that we call preimplantation embryo, or preembryo, or blastocyst, or whatever you want to call it. This is not the head of a pin, this is the point of a pin. And size, of course, doesn't mean everything. I read my daughter, Dr. Seuss, who many of you remember, a person is a person no matter how small. I've said that many times late at night. And I guess my point as a cell biologist is again from the embryology textbooks, that a person is a person no matter how small, but a blastocyst is not a person. And my reason for saying so, and it's based on science. This is not based on philosophy or religion. This is the scientific fact I'll point out, that we know in development 2 individually fertilized eggs can make these little blastocysts, and if they attach in a uterus, about half of the time they will. About half the time they do not, and they die. If they attach, they form a pregnancy, and you can have, of course, fraternal twins. If the embryo splits, however, and it can split in different ways, but if it splits early on, it can again form 2 separate embryos that attach separately. If it attaches a few days after fertilization, it can make a single blastocyst with 2 inner cell masses. How do we know that happened? We know it happens because when the babies are delivered, they'll have one chorion membrane, but they'll each have their own amniotic sac. If the embryo develops as a blastocyst and then forms 2 separate developing humans in the same inner cell mass, that can happen up to 14 days after fertilization. Then the developing identical twins will share the same amnion. And so we can tell that when the babies are delivered. The last little point of biology is sometimes embryos can form independently of one another and fuse together to make a single human, and these are called tetragametic chimeras, meaning they had 4 gametes, 2 sperm and 2 egg that led to their origin. And they're humans that are a mixture of 2 different original embryos. They're normally normal-appearing people, although sometimes they have 2 different pigmentation of their skin, splotchy skin. Or sometimes there'll be a mixture of male and female. These are real people that exist today. So all of these are real case studies from nature, and what they teach us is that a developing embryo up to 14 days has not begun to develop. It has no differentiated cells of any kind. But the point I'm making, that's a well-known phenomenon, but the point I'm making is has not yet committed in regard to individualization. And so if an entity that has not yet committed to individualization, there's at least one documented case where one blastocyst became 4 people, naturally, not through any scientific intervention. Then if it biologically is not yet committed to be an individual, I would argue that in terms of policy and public debate, it doesn't make sense to commit that this is a person, is my point. And of course, the lack of any cell of any kind from the human body would also argue that direction. Well, I'm going to skip over this in the interest of time and just summarize a well-known researcher in reproductive biology was Robert Edwards and, of course, Pat Steptoe, the inventors of in vitro fertilization, who back in the late 70s encountered many of the criticism and debates we have today in modern regenerative medicine. And I want to give credit to these individuals. And the concluding book of their memoirs, A Matter of Life, the last paragraph I'm going to read to us just to close. I'll read the whole thing. Remember, this is 1980. Bob Edwards wrote, "Science moves haphazardly and often unpredictably. Yet what is merely a gleam in the eye of a research scientist today may be familiar to everyone tomorrow. There can be no excuse for manipulating human characteristics unnecessarily, and cloning is an extreme form of manipulation, a needless and unattractive exercise in genetic engineering designed to replace one person by another. But perhaps even this technique, cloning that is, may be utilized eventually for the benefit of humanity in directions which we do not apprehend today, such as averting the rejection of transplanted embryonic cells. Each new discovery should be examined dispassionately and its benefits weighed in the balance. Such is the nature of collaboration between science and medicine." Thank you very much. Yeah, he's going to plug in.
Thank you, Michael. I hope that memoir didn't have any flaws in it, like memoirs, right? We've been hearing about lately. No, I had to come up with a kind of a metaphor for cloning, therapeutic cloning, so I could sort of understand it, because it just defies belief. And the metaphor that I came up with is you take a brand new car and you open up the hood and you grasp the engine and you yank it out, just so there's a bunch of wires and pipes and everything else. Then you take an old car and you open it up and you take the engine out of the old car. No, actually you'd take the whole old car, wouldn't you? You take the whole old car and you just throw it into the hood of the new car that you had just extracted the engine, and close the hood. And not only would the car be able to be driven away, but it would be brand new. That's how I explained it to myself, so I don't know if that's helpful or not. Now we're going to hear from Mr. William Hurlbut, a doctor. That's right. Against my better judgment, I'm going to tell you just a quick little story. Do you mind just a second?
That's fine.
Because I know a little bit about what Dr. Hurlbut has been up against. And this is a very quick story. It's a story about a happening in a church. And just after everyone stood up for the hymns, the pastor happened to see towards the back this very attractive young woman suddenly turn around and wallop the young man behind her. And it sounds like someone's heard this story before. And when everyone was coming out of the church, the pastor saw this gentleman come, who he knew his name was James. He said, and he saw this redness on his cheek, and he said, "James, what happened?" He said, "Well, I was just trying to be nice." He said, "Well, what did you do?" He said, "Well, when we stood up to sing, the woman in front of me, who obviously was wearing this kind of new type of undergarment that made famous here in Washington, actually, and I noticed that her dress was a little bit caught in her gluteal cleft. So I just reached up and just kind of pulled it out for her." And he said, "Well, I guess you understand that you shouldn't have done that." "Oh, absolutely. Now that I know how she feels, I'll never do that again." Well, the next week, he was watching out just after they got up, and that same young woman and the same young man were right behind. And all of a sudden, the young woman turned around and slapped him again. So the pastor thought, well, he hasn't learned his lesson. And in fact, when they came out of the church, and he came and he had his redness on his cheek, he said, "James, what happened? I thought you had learned your lesson." He said, "Oh, I had. I really understood how she felt about this." "Well, what happened?" He said, "Well, she stood up and her dress wasn't caught in her gluteal cleft, so I kind of put it in for her." Well, I don't know whether he's been slapped or not, but he's been making a lot of effort to kind of deal with this issue, this stem cell issue, and to make it so that those people who are against it or those people who are for it would be more conversant of what the people are against it and why, and maybe to deal with some of the reasons that people are against it, etc. It's very, very difficult. I just noticed that we have a distinguished guest has just come in, Senator Orrin Hatch, who actually knows a lot about this issue and feels very, very passionate about it. Senator, would you like to make any comments, or would you like to make a comment after Dr. Hurlbut speaks? Or we would love to hear your thoughts if you feel inclined.
Well, I didn't expect to make any comments here today, but I'm honored to be here with these fine gentlemen, all three of whom I know and know quite well. Robin Cook, of course, a world-class novelist, ophthalmologist, and general surgeon. Mr. Hurlbut, who is one of the top ethicists in this area, and a great professor, and somebody who I spent a considerable amount of time with, who I really respect. And, of course, Michael West, who has been at the cutting edge of this, irritating just about everybody from time to time, but nevertheless very interesting, intelligent, hard-pressing individual who's trying to find ways of helping people to resolve their their difficulties with health care. As you know, we have, maybe I should just spend a second or two on what's happening up in the Congress. As you know, it's been in a big classic battle between the right to life forces, to just mention one group, and those who basically want wide open cloning, and not many of those. And of course, about 6 or 7 different approaches towards this, all of which is caught up in the various political aspects of life. But one thing I'm very pleased to tell you about is after a lot of fighting and a lot of working, we did pass the cord blood bill. Now that was very important because we found some excellent results from cord blood, which still comprises adult stem cells. And that was no small thing. What had happened was the House, when it passed its bill allowing embryonic stem cell work to go forward, it also passed a cord blood bill that wasn't very good. So we, my office in collaboration with a few others, we rewrote the bill. And we wrote it in a way that we could pre-conference it with the House, and we did. We pre-conferenced it with the House, something that's very seldom done anymore. And if it is done, it's kind of miraculous. And we did that all within a week's time. And the House was happy to do that as long as we let them have the number. In other words, the House number, which they deserve, because they had done a tremendous amount of work over in the House and much more than we in the Senate, unfortunately, had been able to do. Of course, we have the filibuster rule. They do not. But almost everybody agreed that we should have the cord blood bill go through. The problem is that we had some on the more liberal side who decided that we wanted to keep that in the overall debate with 6 or 8 various bills and amendments that would be voted on ad seriatim. But what happened is that I was finally, what I did is I went to Bill Frist and I said, look, I want this bill brought up. And then I went to my friend Ted Kennedy and I said, now look, Ted, you clear the decks over here. And he looked at me and said, well, okay. So then Frist brings it up and Tom Harkin objected, who is one of the leaders in the whole stem cell area, along with Dianne Feinstein, Arlen Specter, of course, Ted Kennedy, and others. And he objected. And I went over to talk to him and said, gee, let's get this through. I said, this is something that is a real step in the right direction because we want NIH involved. We do want this as broadly done as we can. And he got kind of irritated with me and said no. Well, the next day, I know that what I had done the day before in getting Kennedy involved, the objection was withdrawn. We passed the cord blood bill, and today that's law. Now, you might say, well, they already are doing that. That's true, but we want full NIH involvement here to set the moral and ethical standards pursuant to which all of this research can be done, but in particular, embryonic stem cell research can be done. Now, we differ, Dr. Hurlbut and I, on how far we should go. But we both have the same concerns. The Hatch-Feinstein bill would prohibit therapeutic cloning or reproductive cloning. It would prohibit reproductive cloning, while allowing the use of embryonic stem cells from the blastocyst to be used in the hope that we might be able to help resolve a lot of mankind's illnesses and problems. And as you know, we have made real headway in some areas with adult stem cells, but it's believed that we can make a lot more difference because of the differentiability that embryonic stem cells appear to have, the ability to be differentiated into, some say, upwards of 220 different forms of human tissue. So I'm just honored to be here. I wanted to come and listen to these 3 excellent people who I know very well, who I believe in, who I know will give you the very best that they have in their minds. And this is probably the most promising medical health care scientific research, as far as I'm concerned, in the history of the world, and that's really saying something. If we are successful in being able to find treatments and or cures, because an effective approach here, kind of supervised by the NIH with the moral and ethical standards laid in place, with a total ban on reproductive cloning, we may be able to help alleviate some of the worst suffering of mankind, and it's worth the effort that all of us are putting in. I don't disparage anybody. I'm pro-life myself, and I do want to make sure that life is protected. But I also want to make sure that little children, like one of our little boys out in Utah was 3 years old when it was discovered that he had the same type of virulent diabetes as his grandfather had, who had suffered for 47 years through 27 operations, losing eyes, members of his body, and so forth, all the way through those years, excruciating pain that whole time. You can imagine the horror that his parents had when they found out that this little boy had the same type of diabetes. I'd like to find some ways of helping children like that, and not just children, but so many others in our society who suffer from maladies that we have within our capacity, at least I believe we have within our capacity, the ability to alleviate. Now, I know that I'm a usurper here, so I just want to thank you for extending me this courtesy, Robin, but also express my high regard for this whole panel, including you. And we can only stay for about an hour, but we're very, very anxious. Now, by the way, I have two of the top people on Capitol Hill with me who really take a real interest in this and who keep me straight on a lot of these issues. So we're very grateful. One is a Robert Wood Johnson Fellow, and the other is Bruce Artem, who headed the Judiciary Committee for me and who has been on loan to me from NIH for over 10 years now. He has done more for NIH than any other person, I think, in the country. But they're also here to learn, and we're so grateful that we've been invited. And thank you for extending this courtesy.
Thank you, Senator. Thank you, Senator, and thank you for those kind words. And I think that we can return them to you as well, that we're really pleased that there are some people in the Senate who are willing to listen rather than just respond kind of reflexly to sort of thoughts or feelings that they might have, but actually listen to people like Dr. William Hurlbut, who really tries to deal with the issues in a way to bring everybody to the table. Dr. Hurlbut?
Thank you, Robin. Thank you, Senator Hatch. I consider it a real honor to be here and a privilege to participate in this important dialogue. This is a very difficult subject and a very difficult debate in our society. This is the cover of recent, about a year ago, edition of the alumni magazine from my medical school, where I was both a student and now am on the teaching faculty. And it shows, as you can see, the divide between the Bible thumper and the scientist. And in this opening remarks on this, the dean of the medical center, Phil Pizzo, wrote, saying a few good things about stem cells. He went on to say, "But today stem cell research remains embattled in the United States. We are once again watching science pitted against political ideology. As a physician, as well as a citizen, I am deeply troubled by what is occurring. It is as if we have entered a time warp and are spectators at the Inquisition's reading of charges against Galileo for his views of the solar system." Is that true? Is that a fair assessment of what's going on? What I want to do today is I want to build on what is said. It's always a little tough being the ethicist around here, because first what happens is all these great promises curing every disease and maybe even death itself, and then you have to come up with the sort of bad news ethics. I want to try to take a positive perspective on that and say that ethics is not some kind of an added extra. Ethics is the glue into which our whole being exists. It's central to our society. It's central to our identity as people. And without the forward progress of science guided by ethics, we don't actually have progress. I recognize that there are important human goods being defended by both sides of this difficult debate. A purely political solution will leave our society bitterly divided. The way this is going is it's eroding our sense of social unity. It's eroding the confidence that a lot of people in this country have concerning the purposes of science and the noble foundations of our science. We don't want to end up with red state medicine, blue state medicine. We don't want to end up with a large number of our fellow citizens entering the hospital and having moral qualms about the foundations on which their therapies have been generated. We don't want the hospital to turn to a place where there is conflict and controversy, what was traditionally a sanctuary of compassionate care and very clear, noble purpose, like those white coats we wear. We don't want this to be a failure, a continuing division. And so in light of that, what I want to do today is I want to describe for you what I think are the fundamental moral issues. And I disagree somewhat with what has been said about the moral status, both the biological and moral status of the human embryo. So I want to explain that. I want to explain what the position of a large number of our fellow citizens is with regard to this issue. And then I want to use the clear and precise definition of the moral problem to show how I think there might be a technological solution that can lead us out of this difficult morass. Now, before I begin this discussion, I want to point out to you that about 6 or 8 months ago now, the President's Council, on which I served, issued a white paper entitled Alternative Sources of Human Pluripotent Stem Cells, pluripotent in this case being a functional identity with embryonic type stem cells. And we put forward 4 basic possible approaches to obtaining these cells without the creation and destruction of human embryos. So I will return to that, but first what I want to do is I want to discuss with you what exactly the moral concerns are in this issue. So as Michael showed us, that is the 8-cell human embryo on the sharp tip of a pin. What are we to make of this entity, these early motions in the unfolding of human biological life? Only from a retrospective view can we recognize the significance of the earliest stages of human life. We are personally grateful that we received the protection and nurture consistent with the full development of our individual being. Yet when looked at through the lens of science, it's evident that an individual human life cannot be described atemporally like a snapshot, but must be recognized in the full procession of continuity and change that is essential for its development. From conception, our unique genetic endowment organizes and guides the expression of our particular nature in its species and individual character. Looked at from one perspective, fertilization initiates the most complex chemical reaction in the known universe. From another perspective, it's the beginning of the central significance of the created world, the embodied emergence of a human person. With regard to fundamental organismal, and from the moral perspective, I'm going to explicate moral inviolability, so organismal initiation of morally specific human existence. With regard to this fundamental transition, the act of fertilization is a leap from zero to everything. The gametes, which are properly understood as kind of instrumental organic agents of the parents, parts of the bodies of the parents, the gametes are not themselves potential life, they are potential causes of conception. The gametes cease to be. They do not unite in the sense of forming a larger whole, but in their interaction and mutual causation, they effect a transition to a new organism, a new human life. In both character and conduct, the zygote, which is the 1-cell human embryo, and by the way, I might add that words are words, of course, but the word embryo, when I was trained in medicine, my dictionary, I still have it, it was a standard dictionary used by almost all medical students at the time, Stedman's. And it says, it defines the embryo as the human organism from fertilization through the end of the 8th week. In both character and conduct, the zygote, the one-celled embryo, and subsequent embryonic stages differ from any other cells or tissues of the body. They contain within themselves the organizing principle of the full human organism. This is not an abstract or hypothetical potential in the sense of a mere possibility. It's not like two chemical reagents sitting on a chemist's shelf that could be mixed. No, these agents, so to speak, are already mixed. This is a reaction in process. It's not mere possibility, but rather potency, an engaged and effective potential in process, an activated dynamic of development in the direction of human fullness of being. Unlike an assembly of parts in which a manufactured product is in no sense present until a completed construction, a living being has a continuous unfolding existence that's inseparable from its emerging form. The form is itself a dynamic process rather than a static structure. In biology, the whole, as the unified organismal principle of growth, precedes and produces the parts. It's this implicit whole with its inherent potency that endows the embryo with its human character, and therefore, from this perspective, its inviolable moral status. To interfere in its development is to transgress upon a life in process. These are some of the stages of this process. As Michael showed you, it forms early on a blastocyst, and of course the hope of embryonic stem cell research is that you can draw on the power of that resource of cells to produce outside of a human body all the cells and tissues for possible therapy. But within the normal natural process of embryogenesis, it divides progressively upwards into increasing expressions of its intrinsic nature. Its complexity unfolds and progresses forward to manifest what it initially only incipiently expresses, but contains in itself as a power, a potency. Now, since Michael talked about some of the issues that are at controversy with regard to the ethical meaning, the ethical argument on this, I just want to take a moment not to add too much science, but I think I should address what are some of the controversies about the nature of this entity that is an embryo or a pre-embryo or blastocyst, whatever you call it. Most of the arguments that this does not have a moral inviolability are based on the idea that there is some kind of a accrued or accumulated quality related to some physical dimension of form or function that then later in development allow you to say, well, there is a human being or human person. One such argument is that until, as I think it was Robin said, until second or third week, he said 21 days, a little later than most people say, but until some point where it manifests visibly some differentiation, that you can't say that there's anything much happening there. It's just like a resource of homogeneous cells. But recent scientific evidence would dispel that notion. The idea that there is no differentiation until you get to this is 21 days roughly, is simply not the case. There are now studies that show that there are distinct cell fates from very early on. There are, at least studies with mice seem to indicate that from the first cell division there's asymmetry, and you can predict the cell fates of these cells, so obviously different. Studies showing certain agents within the cell soup, the cytoplasm of the cells, divide into distinct lineages early on. So there is already a difference between these. And this fact of embryology was documented in a paper in Nature magazine. It's been almost 3 years now. It said, your destiny from day one, the mammalian body plan starts being laid down from the moment of conception, a surprising shift in embryological thinking. The second argument that Michael referred to is the argument of twinning. The idea is that, as he said, as long as the embryo can give rise to more than one individual being, then how can you say that it's already an individual? Yet, ponder the fact that monozygotic twinning, which occurs just once in 240 births, maybe more frequently during gestation, but still infrequently, monozygotic twinning, that's identical twins, does not appear to be either an intrinsic drive or a random process within an embryogenesis. Rather, it's the result of a disruption of normal development by mechanical or biochemical disturbance of fragile cell relationships. This provokes then a kind of splitting of the embryo with a compensatory repair with restitution of two trajectories of embryological development. In considering the implications of twinning for individuation, one might better ask a question from the opposite direction. What keeps each of the cells of the early embryo from becoming a full embryo? Why doesn't it become 10 embryos? Clearly, crucial relational dynamics of position and intercellular communication are already at work, binding that entity together as a single operative unit, unfolding as an organism. From this perspective, twinning is not evidence of the absence of an individual, but of an extraordinary power of compensatory repair that reflects more fully the potency of the individual drive to fullness of form, even at the early stages of embryonic development. A third argument is that until the embryo is implanted, you can't say that it has a potential future. Well, this is a bit of a complicated argument, but in fact, the embryo is fertilized, the fertilization occurs, and the embryo is initiated in the fallopian tubes. It floats down and implants in the uterus beginning around 6 days. And all the way down, it's got a relationship with its mother, and that we emulate it in a dish, but implantation just extends that intimate relationship. Some animals don't ever implant, the non-placental animals. Would we say that they never become organisms? No, of course not. A kangaroo is an organism. The point is that implantation is not, I don't think, a reasonable marker of an ontologic change, a change in the state of being. The intrinsic nature of the entity is that it is a self-developing whole with its drive in the direction of its mature form. And by the way, you might be interested to know that embryos do not have to implant in the uterus. They can implant in any good vascular space. That's why we get abdominal pregnancies. There was even a pregnancy in a woman's liver last year. So there's an intimate relationship, an unfolding human being, intimately related and dependent on its mother all the way through. Now, just to quickly touch on the other argument, a whole array of arguments, and that is function. Most of these arguments are difficult to refute, but also difficult to defend. The idea is that at some specific capacity, then you're a human being. The first and most obvious problem is that the essential function and even the minimal criteria of age of onset are diverse and arbitrarily assigned. Generally, they relate to the onset of something like sentience or awareness of pain or some apparently unique human cognitive capacity like consciousness. This approach is disturbing, though, from an ethical perspective. If human worth is based on actual manifest functions, then does more of that function give an individual a higher moral value? And what are we to make of the parallel functional capacities in animals that we routinely sacrifice for food and medical research? Furthermore, what becomes of human moral status with the degeneration or disappearance of such functions? While we might argue that our relational obligations change along with the changes in function, such as occur with senile dementia, we would not sanction the utilitarian calculus and the purely instrumental use of such people, no matter how promising the medical benefits might be. So this is a complicated problem. These are real ethical issues. These are not religiously based ethical arguments. These are scientifically based and I think reasonable arguments. You may not agree with them, but I think you should acknowledge they are at least reasonable arguments, as reasonable as any other arguments at least. The idea that moral worth is arbitrarily assigned is often said, and if that's true, then everybody's positions are, quote, religious or personal. I think a good argument is that we should honor human life from its initiation as a living organism and hold it separate from all other kinds of things in the world. And that way, if we define it that way, we can then search for an answer that will allow our science to go forward. As I mentioned before, the President's Council took on such a task. I want to say a little word about the President's Council before I proceed. The President's Council is a diverse group of individuals, and we are often accused of being politically appointed. I would say, well, I shouldn't say this in Washington, but if it was intended to be a partisan political appointment, it wasn't the most successful designation of individuals, because we are a very diverse group of people. I, for one, did not have a record of political statement on this. I don't consider myself to have been a political appointee. Nobody asked me when I was interviewed by the White House who I voted for. I did volunteer that I would vote for anybody who was against telemarketing. But when we met and started discussing these issues, it was clear that we were dealing with a very diverse group of perspectives and a very difficult set of issues. And from the very beginning, I was acutely aware, as a physician and the father of a handicapped child, of how important the scientific and medical goals of this. And while I may not agree with everything was said, or my intuitions are maybe different on some things, I largely agree with what Michael said and what Robin said. This is a very exciting time of science. I think a dramatic time. I would almost go along with Senator Hatch. This is the most dramatic moment in medicine. Certainly the convergence of technologies is. The tools are fantastic. And I do not share some of the statements made by conservative opponents of this technology. I do not think we should say, oh, adult stem cells can do everything, therefore we shouldn't look at embryonic stem cells. My feeling about this is we've just put our feet onto Plymouth Rock. We have a whole continent to explore. Why not look at the full range of things? And I also agree with what Michael said. It's not fair to say no therapies yet. That's like saying we've just discovered the quarry, where are the statues? You know, the point is we're getting the tools to understand this. We've sequenced the human genome. We're studying the proteins the genes code for, and now we're on to developmental biology. We are entering a fantastic era of developmental biology. For me, it's very gratifying. When I was an undergraduate major in biology at Stanford, we still were in you've seen the pictures of the quad, the famous pictures of Stanford. The biology department then was still in the quad, and above the door it said zoology. No one says zoology anymore. It was the study of living beings. Now, after almost 100 years of biochemistry and molecular biology, we are returning to the realm of living beings. That's a very exciting moment. It's a quantum leap, as Robin has said, in both possibilities and complexity, but it's also a quantum leap in moral meaning. It means that we need to get this right. We need to find principles that can both guide and protect the very thing we're trying to preserve. We don't want to end up having our cures degrade the very humanity we're trying to heal. So in dealing with this issue in the President's Council, I think we all realized the magnitude of what we were trying to solve. We recognized that across the full span of human existence, there is this amazing unfolding that is uncontroversially related in some way to that which we most highly value. So what is the possibility for finding a way that can preserve those principles, include our whole society, and also go forward? The President's Council put forward 4 proposals. I want to just discuss one of them because I was the author of this proposal. It's called altered nuclear transfer. As described above, natural conception signals the activation of the organizing principle for the self-development and self-maintenance of the full human organism. In the language of stem cell biology, this capacity is termed totipotency, the capacity to form the complete organism. A naturally fertilized egg, the one-celled embryo, is indeed totipotent. In In contrast, a natural embryonic stem cell, or the cells that become embryonic stem cells in the dish, are pluripotent. This term designates the capacity to produce all the cell types of the human body, but not the coherent and integrated unity of a living being. Embryonic stem cells are merely pluripotent. It's a difference between material parts and living wholes. Altered nuclear transfer would draw on the basic techniques of therapeutic cloning, somatic cell nuclear transfer, whatever you want to call it, but with an alteration such that pluripotent stem cells were produced without the creation and destruction of totipotent human embryos. In standard nuclear transfer, or therapeutic cloning, let's jump ahead. In standard nuclear transfer, you all know the nucleus is taken out of an adult cell, placed into an egg that's had its nucleus removed, stimulated to grow. This initiates, at least in some cases when fully constituted, at least in animals, it initiates the beginning of a organismal life. If Dolly was a sheep, then Dolly was once an embryo. With altered nuclear transfer, we could preemptively alter either the genetics or the cytoplasm or both in such a way that we precluded this process of unfolding organism. Could we do this in such a way as to get embryonic stem cells? I think so. There's a natural precedent for this. There's a tumor called a teratoma. Sorry, this is a little gross, but I got to tell you about this. These tumors form something that looks like an embryo, and yet they don't have a trajectory of full development. They end up producing all three primary embryonic cell types, and then later more differentiated cells, and sometimes even parts of bodies, hair, fingernails, and teeth. The white opacities there in that picture are actually teeth inside of a woman's ovary. But these entities have never been considered to be human beings by either the religious tradition or medical tradition. If we could produce such a thing, capable like this of producing embryonic stem cells, we might be able to meet the medical needs and also avoid the ethical problems. Now, I want to stop so that we can have a discussion, but I just want to say a few more things about altered nuclear transfer. There is great realism to a project like this. The idea here is that this would allow us to go forward with consensus. And I believe that if Senator Hatch and others interested in projects like this can get some funding for this, we can go forward and find this. We just need to have something like what Michael was saying last night at dinner, a Manhattan Project, to try to harness the intelligent, constructive, philosophical dialogue with the creative powers of our technology. We can solve this problem. A step in this direction occurred a couple of months ago when Rudolf Janisch at MIT published a paper on altered nuclear transfer showing proof of principle in mice. I guess this slide doesn't show very well, so I won't talk about it. But basically what he did was he showed how the territory in the cell that becomes these cell types can be produced without the full embryo, such that it's reasonable to say, and he's testified to Congress to this effect, that no embryo was produced, and yet he derived fully functional embryonic stem cells from something that was radically different in character from a natural embryo. So this is a hopeful project. There are other dimensions for this project, other ways to do this. There's another altered nuclear transfer is an umbrella concept with many possible avenues of inquiry. Another hopeful dimension of this is called oocyte-assisted reprogramming. You may have read about this, put forward by stem cell biologist Marcus Grumpy at Oregon Health Sciences. So I think we need to get to work as a society and not fight any longer about this. We need to prove what we can do. I'm sorry, I just want to fast forward to some final thoughts on this. The idea is We don't need this huge divide in our society. We need to be a united society. We don't need this kind of stuff either. This is not constructive. We need to use the great resources of this nation, the greatest resource of biomedical technology in the world, to find forward progress. But at the same time, we need to remember that human beings are not just incredible machines. Human beings are that mysterious, unfolding combination of material existence and personal being. Michael pointed out rightly that there is this unfolding organism that has moral worth somewhere along the line. I'm arguing that we should defend it on its full organismal trajectory all the way across from the beginning to the end, draw a fence, draw a border, and use what isn't an organism for advances in science. G.K. Chesterton, the great English author, had a metaphor for this. He said that little boys are playing soccer in a field, and yet right at the edges of the field there are cliffs that go down 2,000 feet. So where are they playing soccer? Nobody wants to do a corner kick. They're all playing in the middle 20 yards. That's the state of our science right now. Because of the concerns about ethics, science is confined to the inner spaces of safe territory. What we need to do now is the hard work of defining with clarity and precision the exact definition of a human organism. That's our boundary. Then we can put a fence up and play in the full field. Some of us want to get the science going, want to get the game going. Others are worried that we're going to fall off the cliff. If we cooperate together, I believe we can find an answer. I just want to read you one thing before I stop. This is a letter that I received from an individual after I testified for the Spectre hearing this summer. I got this in my inbox that night. It said, "Dear Dr. Hurlbut, I enjoyed so much your input on the issue, so forth." She says, "My mother was recently diagnosed with Parkinson's disease at the age of 46. We have been devastated by the news and have since been educating ourselves on all varieties of treatment. My family is Christian, however, so previous stem cell therapies have seemed too unethical." And then she says in an aside, "I've found that this experience has shaken my faith in a major way. I also have hope for the research in adult stem cells, that maybe treatments will be developed that utilize these less questionable cells. The reason I'm writing is that I want you to encourage the President and others to move swiftly in funding this research to alternative methods. I believe this research can greatly aid those with Parkinson's and other heartbreaking diseases. Thank you for your work." Don't we want to have a society that can find a room, space for somebody like that also? Thank you.
Thank you, Dr. Hurlbut. I believe we have some time for some questions for Dr. Hurlbut or Dr. West. Does anybody have any questions?
I wonder if Dr. West might respond to the ANT that Dr. Hurlbut has proposed. From a scientific point of view, what do you think about that process and procedure?
Well, I believe it could be implemented. Is it scientifically a sound concept to implement? I think so. My difference with him on it is this fundamental disagreement as to how we as a society should treat the moral status of a pre-implantation embryo. I see an entity has no cells of the human body, as I said in my presentation, there at all. There's not a single cell from the human body there, and not even yet a commitment on individuation, therefore not, I would argue, biologically a person. I feel that morally we should not, therefore, if we turn this into a therapeutic paradigm, that we should not risk the health of a patient by instituting within it genetic modifications that would make it an altered nuclear transfer, and therefore remove maybe some ethical concerns about that entity, but then risk the health of the patient. Because I believe that the pre-implantation embryo is a proper medical platform for therapy, I think any modification that was not in the patient's interest, if it was simply to address an ethical concern that I believe is a ghost, a phantom, then I think it's not in the patient's interest. And so if it's not in the patient's interest, then I would object to ANT. But could it be implemented? I think so. And could genes be found that could make an altered nuclear transfer protocol work that would not endanger the health of the patient? Potentially. But you see, these things would take years to test. And so I think I would urge us not to do that. On the other hand, if we came down to a national debate and it was on this issue alone, and so all regenerative medicine in the United States would be made illegal, or the choice is altered nuclear transfer. Given that choice, of course, I would choose altered nuclear transfer.
Can I make a comment on that?
Of course.
This proof of principle research was published just this week in Nature magazine by Rudolf Janisch, using a specific gene that he silenced and then brought right back up again. So his opinion was that this would, of course, have to be looked at very carefully, but he thought it could be done safely. I don't think there are necessarily scientific or medical barriers to this. The question is it worth it? Should we have to do it? And I think that is a point that is the point of disagreement here. And yet, even where I'd agree scientifically with Michael, we just have to face the realistic fact that our society is at an impasse. And I want to see the science go forward. I think we should encourage. Some people say as a complementary approach, some people say as the only approach. Well, I don't even want to get into that argument. I'm a doctor. I'm a healer, not a politician or a political scientist. I don't even understand how laws are made. Frankly, all I know is that old saying, never ask what goes into sausages or laws. I put forward this proposal as one little island of unity in a sea of controversy. Couldn't we accept that this might be a positive thing to do now to get this going so that we can open the era of developmental biology? I think just one further point on this, I think if we set this fence at the edge of the soccer field, I think the net gain for science would be greater than the current approach is. I think by clearly and precisely defining the moral boundary as the full organism, we would actually open up projects, many other projects, that would be very positive for science. As it is, I think we're just heading for an endless series of controversies. The ESL, embryonic stem cell research, is just the first.
Question here?
I used to work for the Drug Abuse Administration, and I saw that severe drug abuse, like cocaine and heroin, would affect the neurons in the brain and slow it all down. Do you think that could be a futuristic approach for stem cells to patch it up, maybe, or something with drug abuse too?
Again, with the beginning with the reminder that these are emerging technologies, and it takes a lot of years to make therapies that are at the lab bench available to the hospital bed. The field's called regenerative medicine because embryonic stem cells have very unique properties, some of which are the ability to self-assemble into very complex tissues, like in the central nervous system in the brain. So I think a disproportionately large focus of embryonic stem cell research is in neurobiology. But beyond that, to any particular applications, I wouldn't want to lay out any expectations at this point.
Yes.
I'm Victor Basu, consultant in science and technology policy. This is a question for Dr. West. Replication of cells is considered to be one of the most promising fields for longevity by biogerontologists. Would you amplify on the implications of the stem cell research for longevity, and when do you think it can be extended? And I'm not talking about immortality, but say for 5, 10, 15 years.
Yeah. The a lot of this research actually began in the gerontology research community by scientists such as myself, but that have an interest in the fundamental mechanisms of age-related degenerative disease. I saw Dan Perry here in the audience from the Alliance for Aging Research, who've noticed that a lot of the people who've taken interest in the stem cell debate come from patient advocacy groups in Alzheimer's and Parkinson's disease. And the reason is, of course, that many of the most significant healthcare issues we're facing in the coming decades are an aging population. Increasingly, physicians are seeing older patients, and what they're seeing is degenerative diseases. And if we, as a medical researcher, put those tissues under a microscope and see what are the primary mechanisms of so many of these age-related degenerative diseases, we see arrows pointing to common mechanisms. And so embryonic stem cell technology, as you may be indicating, was largely invented as a new technology platform to address age-related degenerative diseases, but of course have many applications in the diseases that young people have as well. In regard to timelines, the applications in age-related degenerative diseases, I think, is no different than any other. Currently, researchers are learning to turn embryonic stem cells into many medically useful cell types. Biotech companies such as our own are working out standard operating procedures for differentiating and manufacturing cells, and are working up plans for approaching the FDA for the beginnings of human clinical trials, which, depending on the disease, can take from 3 to up to 7 years sometimes for a clinical study. So, I would say that the end of this decade we'll see the first applications of embryonic stem cells in the clinic.
This is a question for Dr. Hurlbut. Right now we have no embryonic stem cell lines that are developed from nuclear transfer procedures with the collapse of what happened in Korea, but it appears that Dr. Huang's lab was successful in generating an embryonic stem cell line from a parthenode. And so I'm wondering if that is a promising technology that would avoid these moral qualms.
Yeah, that's a great question. I think that's exactly the kind of question we're going to face. Parthenogenesis. Michael was saying last night how we'll eventually create stem cell type, we'll differentiate them out into the next branching layers and reaggregate them. So we're going to face laboratory creation of human parts apart from the whole. It's the same question that altered nuclear transfer asks. Is parthenogenesis producing a human organism? Is reaggregation at some level producing a human organism? Below that threshold, not. That's why I think we need to define with clarity and precision exactly what the moral principles are trying to defend. I have looked at that question of parthenogenesis. There's a Dominican priest who is writing on that and argues that parthenogenesis should be considered to be a non-organism producing process. And I can go pretty far with that. I think he might be right about that. And the trouble is that parthenogenesis in varied mammalian species has a different measure of completion and may be different from genome to genome, and so it's a little tricky. The principle, I think, could be equally met with altered nuclear transfer by having a sure preemptive alteration that would prevent getting even as far as parthenogenetic entities do. In other words, parthenogenesis is not the ideal way to get these cells anyway, because you're limited to fertile female genomes, and there are various disadvantages. Also, parthenogenetic genomes that I don't get into scientific complexity, but they have a lot more homozygosity. A point I want to make is a little technical bit of a point, but Michael said that that altered nuclear transfer might have medical dangers associated with it. And I said afterwards that I didn't think that was, I think we could probably overcome that. But we should be aware that when we take embryonic stem cell lines from IVF-produced embryos, in vitro fertilization embryos at the 4 to 5 day stage, there's something we are not paying attention to when we talk about that. The genetics in those cells has never been proven to form an organism. It's a new combination because of, in the production of gametes, there's reshuffling of the genes. There's a statistically at least a ongoing rate of mutation. We don't actually know for sure that those cells can do safely what we're projecting them to do. We might be able to study that too, but I'm just pointing out that this whole field is full of potential pitfalls. Therapeutic cloning, so-called, or renamed by the President's Council, cloning for biomedical research, also involves potential medical problems, epigenetic problems. Now, Rudolf Janisch has done studies at MIT that seem to indicate that that isn't a problem, but how fine an analysis has he done? We'll have to wait and see. The point is that at every level we need to do the hard work scientifically, but also the hard work morally to define what's both possible and moral.
The question here?
I'd like to know what the impact of the Korean disaster has been, because I've read it both ways, that it's both set this field back several decades and that in fact it has now given companies like yours, Dr. West, a new lease on life. What's the
Well, I think it's, of course, an unfortunate and sad event, but I think the impact it has is what impact we want it to have. Obviously, the opponents of stem cell research have tried to make new newspaper advertisements and a new effort for a campaign. But you know, it's an unfortunate event in science that has occurred in the past. But I think its impact is negligible, unless we want to make it something other than that.
I have a newspaper article from a few weeks before the scandal. It says, he's shown here with Snuppy the dog he cloned. It says, human stem cell ethics not going to the dogs. Well, we found out it was. No, I don't think it is. I mean, you know, it's a great question. I think it's going to have a big impact on social acceptance of this. It's already a field besmirched by controversy, and I think this will be a setback for it. I think, nonetheless, I think that there is a unique opportunity here. I agree with Michael. This is a chance for our nation to harness its research potential and its wisdom, collective wisdom, to do this right. In other words, we've actually been given a chance to rewrite the history of this. I'd like to see it rewritten through alternative methods and strongly promoted. I don't see how anybody could oppose that, frankly. It seems mean-spirited not to find an answer that all of America could live with.
Can I resonate with that and agree with you wholeheartedly? The one aspect of this unfortunate event that has occurred there is does indeed offer an opportunity for the United States to take the lead again in this area, which has been newspaper headlines recently said we've been losing. And I think it's a moral obligation for us to do so. We've been all appealing a lot, I think, to the problems of individuals afflicted with disease, but you know, those in medical research, we think about these things. And I remember one similar discussion to this, which I participated in the United Kingdom, where there's been a lot of thoughtful debate on this. And on the panel was a member of the Master of the Guild of Catholic Doctors in the United Kingdom. And one of the people in the audience, a member of the Royal Society, asked that physician the question: we have, on one hand, human preimplantation embryo, and the other hand, the life of a young girl dying of leukemia and the need of ES-derived cells to save her life. Which death do you hate worse? I remember is the way the question was put. And after obfuscating, the physician finally said, I would choose the death of the little girl. Because ultimately, life is short, and our lives here are a small thing. compared to eternity and the moral choices we make. And I realized in that conversation I had a very fundamental disagreement on this. In my mind, and maybe flip is an inappropriate description, but this quick, nevertheless, decision to choose the death of a child, to me is just, it raised a hair on the back of my neck. In my mind, we as the United States have a really unique opportunity with the technology and power that we possess, and I think the moral courage to make really intelligent and carefully thought out moral decisions. I think, again, we have another historic opportunity now to lead the world in this area. And I think that moral decision that we have before us, on the one hand, the pre-implantation embryo, the other, the lives of real people who need medical therapies, I think the sobriety that we need to take to this decision is immense. I think it ought to be a very thoughtful decision. And whether it be altered nuclear transfer or I think simple nuclear transfer, I think we have a historic opportunity to lead the world in this area now, and I think we should take that opportunity and run with it.
I'd like to dispute the moral analysis that you gave of the British case, because in one case, the death is the result of natural causes. In the other case, it's the intentional destruction. And that is what makes it morally significant. And here I turn to Dr. Hurlbut. It seems to me that what you're proposing, if I understood it correctly, is to set the fence, as you call it, at the point of intentional destruction of human embryos, right? And then the crucial question becomes, well, when do we have a human embryo? And that's the challenge that we're facing right now, and it is, how do we establish both conceptually and empirically when we have a human embryo? And if we do, then that's what should be respected, if I take it.
My point in my presentation was we have to make moral choices in this world, and many of us feel we're held accountable for those. And the presence of identical twinning in our midst, and other examples I gave, I think are a clear indication that the pre-implantation embryo is not an organismic human, in the sense that identical twins cannot trace the beginnings of their life at fertilization. They clearly, as I showed, there's clear biological evidence that many identical twins can trace the date of their origin as an individual human being up to day 14, which is a week after where we derive these stem cells.
Why don't you follow up?
Yes, follow up.
Yeah. Well, the twinning argument has been extensively discussed in philosophical circles, of course, and the problem really is, how do you characterize the possibility of twinning? One way of doing it is by saying, well, the fact that something can divide does not logically entail that it actually is not one. I mean, that happens in plants, for instance. No one would dispute that a certain plant is a single organism, and yet you can get two plants from it. So there's an alternative analysis of the twinning argument, which holds that indeed for most people, all of us who do not have a twin brother or sister, for all of us, we can trace our origin to the zygote. In the case of twinning, then, we have to give an ad hoc explanation for a very unusual biological event, but that does not necessarily entail that there was no individual there before the event.
Obviously, we could talk numerous weeks on this issue. There's a very good book written by a Catholic theologian, Ford, I think, is it Norman Ford? Norman. Where he carefully thought through the facts of the matter, I think dispassionately, and wrote a very nice monograph on it. What's it called? How Life Begins, or When Human Life Begins, by Norman Ford. When did I begin to exist? You know the book. So, and I would point out, came to the conclusion that the beginnings of human life, from his perspective and studying, that should be considered 14 days as well.
I've talked with Norman Ford, and I think he's somewhat modified his views on that recently. He certainly is in agreement with the his Catholic tradition that you should honor life and avoid all risk of violating life at that stage.
As I understand it, because of the formal position of the Vatican, he's agreed with the Vatican position.
I'm sorry to take so much time, but what happens with the writings of Norman Ford is that they were drafted and published long before the new evidence of the sort that Dr. Hurlbut presented was known. In other words, this idea that there's differentiation from the first cleavage onwards was totally unknown to Norman Ford. But now we have to base it on the most recent scientific evidence, not on what the Vatican says.
Yeah, but I would urge that the evidence actually swings my way. The evidence that Dr. Hurlbut was referring to is that there's a polarity in the early embryo, sort of a head-tail polarity. But that polarity exists in the egg cell prior to fertilization. So to say that there's a polarity that exists in the egg cell, well, we don't say the egg cell is a human being. That property alone, I don't think, should cause us to conclude that aggregation of cells, the pre-implantation embryos, is a human being because it has a polarity that pre-exists the fertilization event itself.
Oh no, it's not polarity that makes the entity a human being. That's evidence that there is differentiation from the beginning. That's all. And you were claiming that it was just a homogeneous clump of cells without a differentiated trajectories, and that's biologically not true.
The evidence I've seen originates from amphibian, not mammalian species, from amphibian, mostly from frog studies, where differentiation occurs very early on.
Very what? Very early?
Very. A lot of the evidence I've seen presented that there is the beginnings of development, and the very early embryo comes from non-mammalian species, from frog species.
Well, I was citing work on mice done by Magdalena Zuniga-Gertz in England, very well respected developmental biologist. So this does appear to be the case with mammalian species. And the slides I showed, the one that had the cells with a yellow marker in the middle, showing that it was an asymmetric distribution of cell contents, those were human cells.
The biology we're referring to is important in this debate. It's called regulative development. The fact that human and many other mammalian species, they accumulate a cluster of cells, of raw material, from which then development will proceed, rather than having development proceed very early on. And so this is what allows the early human embryo or the early many other mammalian embryos to divide up, this cluster of cells to grow up to 14 days, and then development commence, and up to 4 individuals coming from the same embryo. It's a well-known piece of biology called regulative development.
Let's have one more question.
You've talked about the various controversies about the sources of these cells, and I just wonder if there are any. I know there are, but what are the potential controversies about uses of these cells down the road?
I'm sorry, I didn't quite understand. What are the potential uses that we might?
You've talked about all the wonderful uses, potential uses of these cells down the road, and I just wonder: are there other uses that we might in the future find more troubling?
More troubling?
More problematic.
Oh, I thought you were going to say more other positive uses. Let me quickly throw out 2, and then let Dr. Hurlbut answer as well. Besides the use in cell therapy, there's a lot of enthusiasm in the pharmaceutical industry about being able to, as I mentioned, the power of these cells is not only being able to make any cell of the human body, but genetically engineered in any way. For the first time, this confluence of DNA technology with cell biology. This, of course, has medical application in cell therapy, but also the pharmaceutical industry is very interested in being able to make genetically engineered human cells of any kind, put them in the laboratory dish, and then test drugs on them, so to be able to potentially find new drugs for the treatment of disease. In terms of problematic aspects, it's ironic. the enormous debate about the origins of embryonic stem cells, it doesn't trouble me. I think you can tell that based on my considerations of this area. However, I can see other areas that do gravely concern me. The ability to genetically modify in any way these cells could eventually lead to the people would want to genetically engineer human beings in any way. And you've seen in the newspapers just a few weeks ago the fluorescent pigs made in another country. Well, fluorescent pigs don't bother me, but the concept of beginning to genetically engineer the human being is, in a sense, playing God with human life, actual human beings. And these kinds of technologies could be adapted to such ends. You can, Dr. Cook could write better or paint better stories or possibilities of how that could be abused than me. He has more of an imagination than I do in that regard. But those are areas where I think, the intent is not necessarily to cure diabetes or Parkinson's disease or rebuild the heart after a myocardial infarct. The goal is to, and we hear discussion about this, enhance human characteristics. There are thoughtful people here in the United States that are saying, well, we're going to be doing this. If your children are going to be competing with other children in the school and they're genetically engineered to have enhanced memory, you're going to want your children to have that too. And those are ethical debates that I think are worthy of discussion. I don't think it's imminent, but those trouble me as an individual.
I'll add only to that there are, mostly it's not controversial, and mostly it's very positive, and I'm very personally excited about what can be done with these cells. But I would add just one little layer of concern that some people have raised. We will be able to, perhaps, looks likely, at some point, produce cells and combinations of cells that are troublingly ambiguous in their moral meaning. For some people, the idea that we could generate human eggs from embryonic stem cells is a troubling prospect. Some people consider eggs to be somewhat sacred themselves. I don't personally share that concern, but I do think we could violate some basic notions of human dignity by generating human parts and using them in a disrespectful way. I think the only thing that justifies this kind of invasion into this territory of central significance in human existence, like the use of human gametes, is the seriousness of the purposes. So frivolous purposes could be used. Also, we could combine elements, as we were saying earlier, and produce advanced tissues and maybe organs, or maybe even organ systems someday. Let's look well in advance. So how much brain tissue in one sensory modality would be acceptable? It all points to the fact that we need to define with clarity and precision that which is our moral boundary. I think that there's a serious debate going on here. It's not just some ignorant religious people debating some well-informed scientists. I have a pretty good scientific background myself, and yet I feel the weight of these moral issues. I would just since this is probably the last thing I'll get to say, I just want to say one last thing here. In 1999, President Clinton, this is not President Bush, okay, President Clinton's National Bioethics Advisory Commission issued a report entitled Ethical Issues in Human Stem Cell Research. In that report, they acknowledged that a week-old human embryo is a form of human life that deserves respect. The commission stated, "In our judgment, the derivation of stem cells from embryos remaining following infertility treatments is justifiable only if no less morally problematic alternatives are available for advancing the research." I think we have a moral obligation to seek a solution that is uncontroversial, does not violate the moral sensibilities of a large percentage of our population. If we do it that way, I think biomedical science can go forward as a part of our national identity as a noble and progressive society. That kind of a solution would be in keeping with the American spirit and would be a triumph for humanity as a whole?
Well, I think that's a wonderful place to stop, and I think everyone here would agree that this little forum was aptly named, and I'd like to personally and for the Woodrow Wilson Center thank these two doctors, Dr. Michael West and Dr. William Hurlbut, for coming here and entertaining us. And the last thing, since I have the last word, I'd like to say that novelists like myself, we do tend to kind of grab on the bad side, but I'm going to say that the reason we do it is so that we can have the public become aware of these issues. I don't do it just to scare everybody. Thank you very much for coming.